MCQ. Any part of a plant taken out and grown in a test tube under sterile conditions in special nutrient media is called as dash. Options are tissue, explant, implant, organ. And the answer is option B, explant. The capacity to generate a whole plant from any explant is called as dash. Options are hybridization, tissue culture, implantation, totipotency. And the answer is option D, totipotency. The method of producing thousands of plants through tissue culture is called as dash. Option A says micropropagation, option B implantation, option C explant, option D somaclones. And the option is A, micropropagation. All the MCQs are 1 mark. Similarly, assertion, reason type questions also carry 1 mark. Assertion: each of the plants produced by micropropagation are somaclones. Reason: they will be genetically identical to the original plant from which they were grown. So what could be its answer? And the answer is that both assertion as well as reason are true, and reason is the correct explanation of assertion. Now this question carries 2 marks. Tissue culture can recover healthy plants from diseased plants. Explain how. Give any 2 examples. Carries 2 marks. Answer is, even if the plant is infected with a virus, the meristem, that can be apical meristem or axillary meristem, they are free of virus. Hence, one can remove the meristem and grow it in vitro in order to obtain virus-free plants. And examples are banana, sugarcane. They ask for 2 examples of this, so you are writing banana and sugarcane, which is there in the textbook. Next question is, what is the difference between somaclones and somatic hybrids? It carries 2 marks. Answer will be, when a whole plant is grown from any cell of a plant, it's called as somaclone. While plants grown from protoplasts of different varieties of a plant, then they are called as somatic hybrids. Somaclones will be genetically identical to the parent plant, whereas somatic hybrids will have genes of the varieties used to create them. Now this is a case study type question carrying 4 marks. Genetically modified organisms are living organisms, plants, animals, or microorganisms whose genetic material has been artificially altered using genetic engineering techniques. This process introduces specific traits such as pesticide resistance, faster growth, enhanced nutrition, which do not occur naturally. Genetically modified organisms are extensively used in agriculture and medicine. Now the questions are like this. What are genetically modified organisms? Carries 1 mark. List any 2 uses of genetically modified organisms. So since they are asking any 2 uses, they will carry 2 marks. C part of question, give an example to show that genetically modified organisms improve nutritional value of food. That carries 1 mark. First part of question, what are genetically modified organisms? The answer is plants, bacteria, fungi and animals whose genes have been altered or changed are called genetically modified organisms. B part of question, list any 2 uses of genetically modified organisms. One use is genetically modified organisms have made crops more tolerant to abiotic stresses like cold, drought, salt, heat. Second kind of use is they have reduced reliance on chemical pesticides, like for example pest resistant crops. Now the C part of question, give an example to show that genetically modified organisms improve nutritional value of food. That carries 1 mark. Example is golden rice, which is vitamin A enriched rice. So totally this question carries 4 marks. Next question carries 2 marks, and that is, what is the mechanism behind Bt cotton's resistance? The answer goes like this. The bacterium Bacillus thuringiensis produces inactive protoxins, which when ingested by the insect are converted to active toxin due to the alkaline pH of the insect gut. The toxin binds to the surface epithelial cells, causing lysis and death of the pest. Now next is a 1 mark multiple choice question. The proteins encoded by which cry gene control corn borer? Option A, cry1AC, option B, cry2AB, option C, cry1AB, and option D, cry2AC. And the answer is option C, cry1AB. Next is a 3 marks question. A nematode Meloidegyne incognitia infects the roots of tobacco plants, significantly reducing yield. Based on the mechanism of RNA interference, how can scientists use this technology to protect the plants? So the answer would be like this. Using agrobacterium vectors, double-stranded RNA was introduced into the plant. These silenced the specific mRNA of the nematode. So the parasite could not survive in a transgenic host, expressing specific interfering RNA. The transgenic plant therefore got itself protected from the parasite. So this is an assertion recent question carrying 1 mark. Assertion: RNA interference is used to protect plants against nematode infestation. Reason: RNA interference method involves silencing of a specific mRNA by a complementary double-stranded RNA molecule that binds to and prevents the translation of the mRNA. Suppose the answer both assertion and reason are true, and R is the correct explanation of A. That means reason is the correct explanation of assertion. Next question is a 2 marks one based on the topic of RNA interference. Question goes like this. What is the source of the complementary RNA that causes nematode mRNA silencing in tobacco plants? The source is from an infection by viruses having RNA genomes or mobile genetic elements called transposons that replicate via an RNA intermediate. Question is a multiple choice question for carrying 1 mark. Name the nematode that infects the roots of tobacco plants, reducing its yield. So they are all nematodes, but we have to choose a nematode that infects the roots of tobacco plants. Option A, Ascaris. Option B, Meloidegyne incognitia. Option C, Wuchereria. Option D, Ankylostoma. And the answer is option B, Meloidegyne incognitia. This question is a multiple choice one. Nematode-specific genes were introduced into the host tobacco plants using which of the following vectors? So it carries 1 mark. Options are Bacillus thuringiensis, option B, E. coli, option C, lactobacillus, option D, agrobacteria. Which hormone is used for diabetes? It isn't advisable to extract this hormone from pancreas of slaughtered cattle and pigs. Give reason. This question carries 2 marks. The answer, which hormone is used for diabetes, is insulin. And the second part of the question, why it isn't advisable to extract the hormone from pancreas of slaughtered cattle and pigs? That's because it causes allergy in the patients. So the answer is option D, agrobacterium. So nematode-specific genes were introduced into the host tobacco plants using agrobacterium vectors. Previously, insulin for diabetes was extracted from the pancreas of slaughtered cattle and pigs, causing severe allergic reactions in some patients. Why did this happen? It happened because they were not human proteins. They were proteins of the cattle. That's why the allergy happened. What is option B? How does genetically engineered insulin overcome this? That is also carrying 1 mark. So genetically engineered insulin has human protein. So it can overcome this problem of allergy. Option C says, what are the two polypeptide chains present in insulin? Polypeptide A, polypeptide B. C peptide will be removed. Option D, what is a major challenge in producing this in bacteria? And how do scientists solve it? This carries 2 marks. So the problem is that bacteria is not able to remove the C peptide of proinsulin. If the C peptide is removed only, proinsulin will become mature insulin. Now how did that company help? Eli Lilly, an American company, prepared 2 DNA sequences, A and D chains of human insulin, and introduced them in the plasmids of E. coli. So they synthesized A chain and B chain separately. Then the chains A and B were produced separately, extracted and combined by creating disulfide bonds to form human insulin. So at the end they combined both the chains. So this was a long answer question carrying 5 marks. A 4-year-old girl is diagnosed with severe combined immunodeficiency due to a deficiency of the enzyme adenosine deaminase. This enzyme is crucial for the immune system to function. The disease is caused by a deletion in the gene for adenosine deaminase. Conventional treatments like bone marrow transplantation have not been successful. Doctors decide to attempt a gene therapy. Now we will get the questions related to this. So it's a case study type of question. The first question is, what is a gene therapy? Carries 1 mark. The answer is, genes are inserted into a person's cells and tissues to treat a disease. So that is called as gene therapy. The second part of question B is, how are lymphocytes used for adenosine deaminase gene therapy? And why is it not completely curative? Carries 2 marks. So adenosine deaminase complementary DNA using a retroviral vector is first introduced into lymphocytes. And lymphocytes will be cultured outside the body. Then they will be returned back to the patient. But they are not completely curative, as lymphocytes are not immortal. Means lymphocytes will die. So they need to be periodically infused into the patient. C part of question is, how can it be a permanent cure then? Carries 1 mark. If the gene isolate from marrow cells producing adenosine deaminase is introduced into the cells at early embryonic stages, then it could be a permanent cure. So it's a 4-mark question, case study based. A patient is suspected of having a very early stage HIV infection, but traditional antibody based tests come back negative. Which molecular diagnostic tool is most suitable for detecting the virus at this stage, and why? Carries 2 marks. Very low concentration of a bacteria or virus at a time when the symptoms of the disease are not yet visible can be detected by the amplification of the nucleic acid by PCR. So the molecular diagnostic tool that is suitable for detecting the virus at the early stages, PCR. How does a single-stranded DNA or RNA probe tagged with a radioactive molecule help in identifying a genetic disorder? This question carries 2 marks. So the answer will be a single-stranded DNA or RNA tagged with a radioactive molecule, which is called as probe, is allowed to hybridize to its complementary DNA in a clone of cells, followed by detection using autoradiography. The clone having the mutated gene will hence not appear on the photographic film because the probe will not have a complementarity with the mutated gene in that. So that is how we identify a genetic disorder. Now this is an MCQ. ELISA is based on the principle of dash, carries 1 mark. ELISA means what? Enzyme linked immunosorbent assay. So options are option A, complementary hybridization of nucleic acids. Option B, antigen antibody reactions. Option C, gel electrophoresis. Option D, polymerase chain reaction. Answer is option B, antigen antibody reactions. ELISA test is based on the principle of antigen antibody reactions. Now this is a short answer type question carrying 3 marks. Give a reason why transgenic animals are used for chemical safety testing. Carries 1 mark. Answer is as they are more sensitive to toxic substances than non-transgenic animals. That's the answer. Option B, which human protein is used to treat emphysema? Carries 1 mark. The human protein is alpha-1 antitrypsin. Option C, the first transgenic cow, Rosie, produced what human protein in its milk? Carries 1 mark. Human protein's name is human alpha-lactalbumin. Now this is a multiple choice question. The Indian government has set up organizations such as GEAC, which will make decisions regarding the validity of GM research and the safety of introducing genetically modified organisms for public services. What does GEAC stand for? Option A says Genetic Engineering Approval Committee. Option B says Genome Engineering Action Committee. Option C says Genetic Environmental Approval Committee. Option D says Genuine Engineering Action Committee. So what is the correct answer? The answer is option A, Genetic Engineering Approval Committee. That is in the textbook. Nowadays it's also called as Genetic Engineering Appraisal Committee. Hey, thank you children.